we had gotten to exceptions to chromosome theory, right? So we had talked about the fact that genes are found on chromosomes, right? You inherit kind of all the information that makes you you via chromosomes, right? That are shuffled around and distributed in meiosis. And then we had talked about sex-linked genes and the fact that sex-linked genes act a little differently, right? Because they're attached to the X chromosome. So like if you have two X chromosomes, you kind of, well, it acts more like a regular chromosome versus if you have an X and a Y chromosome, which don't really match up. And then you can have recessive traits on that X chromosome that kind of act like their dominant traits. And then, well, we talked about dosage compensation, because if you have two X chromosomes, you don't want to make double of everything. So you kind of turn one off, right? So we'd worked through all that. Now, as with kind of everything, right, we talk about kind of the nice, simple, quote, simple, straightforward rules, and then we get to some exceptions where things are a little weird. So we're going to talk about exceptions to chromosomal theory. There's actually not a whole lot to it. Generally speaking, your chromosomes act like we expect them to. But we've got some DNA that behaves a little differently, right? So all the way back to endosymbiotic theory that happened in a chapter that was way earlier in the book, right? What did endosymbiotic theory state? What is endosymbiotic theory? It is exactly that thing, yeah. So endosymbiotic theory was that idea that like things in our cells, like mitochondria, were initially, right, free living little things that our little single-celled ancestors engulfed, for some reason didn't eat, kind of used for their metabolic abilities, right? And so mitochondria and chloroplast were initially free living prokaryotes that got engulfed and eventually became symbiotic partners, eventually, right, kind of non-optional, right? We're basically all kind of rolled up into the same organism more or less at this point. So question one, how dark does it get if I turn this other light off? Meh, that's not too bad. So what is something unique then about these organelles that's going to affect how DNA is being inherited? Oh, they could, but your nuclear DNA is mutating. We're in the, like your mitochondrial DNA, right? Your DNA has its own mitochondria, separate from your nuclear DNA, right? And everything we've talked about so far, Mendel and meiosis and all this stuff, has been talking specifically about the DNA in your nucleus, right? It does. It controls, right? Yeah, the mitochondria has its own DNA that's doing the mitochondria stuff, right? And so if it's not in the nucleus, what is not happening to it? What happens to the what happens to the DNA in your nucleus that you inherit? What happens to the DNA in the germline cells that is not happening to your mitochondria?
It's set to the side.
Not mitosis. Well, actually both, but yeah. Yeah, it's not partitioned during cell division, right? Your mitochondria don't duplicate, assort, cross over, and split or anything like that. None of that is happening, meiosis or mitosis, right? Your mitochondria are actually still just acting like little prokaryotes. They're just doing binary fission every time the cells divide, right? So remember when we first talked about mitosis, we spent kind of a half a minute talking about binary fission where they just have a little one single ring chromosome. They make a copy, they split in half, job done, right? And so that's what your mitochondria are doing, right? So when mitosis is about to happen, or since we're talking about inheritance, meiosis is about to happen, your mitochondria just do a bunch of binary fission, make doubles, partition them into the cytoplasm, right, and kind of send them each on their way. No independent assortment, no crossing over, none of that kind of stuff, right? And they are specifically portioned into the cytoplasm of egg cells, right? Sperm cells are super tiny. Basically, sperm cells only carry nuclear DNA. They got nothing else. That's all they've got. They just have the information. That's it. Your egg cells are much larger. They're like the largest cells you've got, basically. So they basically have everything in them, right? They've got their haploid copy of all the DNA. They have mitochondria in them. And because of that, all your mitochondrial DNA actually only comes from your mom. So anything coded for by the DNA in your mitochondria, you only inherit from your mother, right? Which means that if we're looking at inheritance and traits, right? And these are things that are going to probably have more to do with kind of metabolism and that sort of thing, because it's what the mitochondria is doing. But things that are happening in the mitochondrial DNA are only going to be inherited maternally. If your father has some sort of mitochondrial DNA disorder or mutation, doesn't matter. You can't get it. Right? So we call that maternal inheritance. Right? Chloroplasts in plants work basically the same way. Plants are weird and they just have exceptions all over the place. Generally, it's maternally inherited, right? But some of them will do weird things where it's biparental or paternal, which is extra weird. But generally, chloroplasts kind of work just like our mitochondria do. They inherit it from their mother, which is weird in a plant, the female part of the plant. All right, so we're going to very briefly, we're going to kind of gloss over genetic mapping. Sounds like a pain. It is kind of a pain. It can get really complicated. We're going to skip the most complicated bits of it. Your book goes like way into more detail than people who do genetic mapping care about. Personal opinion, it's fine. So we're going to talk about segregation of genes on chromosomes. Basically, right, just this idea that now that we know that your traits are carried on chromosomes in genes, and those genes kind of occur at predictable places on the chromosomes, you can then map where things are on chromosomes, right, and then predict inheritance, recombination, all this other kind of stuff, right? And then if you're wanting to track some trait, some mutation, you can actually know exactly where it is in somebody's genome, which can be a handy thing. So, bless you. Of Mendel's 7 traits, 6 of them are on different chromosomes. This is just more reasons Mendel was just ridiculously lucky, right? 6 of them were on separate chromosomes. And so, you know, he found logically that traits act independently because of independent assortment. It's easy when they're all on separate chromosomes. But one of them wasn't. Doesn't matter which one it was, but one of them was actually kind of doubled up with one of the other 6. But he still found that they all, right, that all 7 of them assorted independently. How is that possible? So there's independent assortment, right, where each chromosome kind of gets shuffled between the cells. But if you've got 2 traits on the same chromosome, why would it still look like those 2 traits assort independently? What else is happening during meiosis? It's on the screen. Crossing over happens during meiosis, and will also move some genes around, right? And so actually, you generally have more genes that assort independently than you have chromosomes. It's not like there are only 23 genes that assort independently in a human, right? That's not true. So back to meiosis, always. So in prophase 1, crossing over happens, right? So, right, this is that same kind of series of crosses, right? We've got a parent generation with a true breeding recessive and a true breeding homozygous dominant, right? We make this F1 generation where one chromosome has both recessive traits, one chromosome has both dominant traits, right? And so without crossing over, right, these individuals go through meiosis and pass on what we call the parental chromosomes, right? where all the gametes are either homozygous recessive, just like that chromosome, or homozygous dominant, just like that chromosome, right? And so these two recessive genes, without crossing over, would never sort independently, right? They just go together. They just kind of end up in the same place. But because crossing over occurs, right now we swap, say, this dominant trait B for this recessive trait B. Now, because crossing over occurs, we've got all four of those choices we saw in the Punnett square, right? If this gets passed on, we've got a homozygous recessive individual. We've got our two heterozygous options, which we call recombinant because the result of recombination crossing over. And then this homozygous dominant individual, right? And these gametes match the parents, so we call them parental. The ones that undergo crossing over, we call recombinant. And it makes it look like, even though those genes are on the same chromosome, they can still get kind of split up and shuffled around, right? Because crossing over happens, right? Yeah, you can also call crossing over recombination because it's like a recombination of alleles. So we call them recombinant chromosomes. So crossing over results in recombinant chromosomes. All right. So if we want to know, so they keep saying genetic maps or genetic mapping in the textbook. They just mean like figuring out where the genes are on your chromosome, right? That's all we're doing. And we can kind of predict this by looking at the way that different genes behave, right? So we can map the locations of genes on a chromosome looking at the rates of recombination, right? The frequency, the amount of crossing over that happens between a couple of genes is related to how far apart they are on the chromosome, which kind of makes sense, right? So if I've got two genes, gene A and gene B, and they're really far apart on the chromosome, then any crossing over that happens like in the whole length of the chromosome will swap them out, right? Because they're really far apart. Versus if they're really close together on the chromosome, then the only way these two genes are sorted independently is if crossing over happens like specifically here, right, in the DNA molecule. It's just less likely, right? So it's just kind of an odd sort of thing, right? So as physical distance on a chromosome increases, so does the probability of recombination or crossing over. So if you're looking at things and you see how often, right, these genes kind of cross over and get swapped around, you can predict how far apart they are on the chromosome. All right. So that's just kind of the relationship between crossing over and mapping genes onto chromosomes. Now, if they are close together, right, recombination or crossing over is not going to happen very often, right? And so you are more likely to see the parental types, right? Because you're not going to get those recombinant chromosomes very often. You're mostly just going to be passing on, right, these two genes, these two traits together. And so we say they're linked, right? If your DNA is a long kind of train of genes, these linked genes are kind of like two train cars that are connected to each other. There we go, right? And so they tend to just be inherited together because it's really unlikely that the swap is going to happen specifically between these two train cars. Versus if you have one gene that's like up at the front of the train and one that's at the back, right? So that any shuffling that happens in between is going to kind of make them separate and act independently, right? So we can have linked genes. Basically just means they're really close together on the chromosome. All right, that's enough mapping. So let's look at kind of like what this actually looks like. So looking at human genetic disorders, which is kind of a really pertinent and practical end of studying inheritance and the behavior of genes and where things are on chromosomes and how likely or unlikely, right, traits are to be passed on, right? There's obviously a huge field which studies specifically human genetic disorders, frequently focusing right on single genetic disorders. So we're going to talk about mutations and non-disjunction, and a little bit on epigenetics, just because it's kind of fun and interesting, this genetic imprinting idea. But human genetic disorders are genetic, so they're heritable, right? They run in families. And so this is just where, right, mutations in DNA, where DNA gets changed, right? DNA codes for proteins. So if there's a mutation, if something changes in your DNA, it can change the shape of the protein, right? And if we remember all the way back to proteins, if you change the shape of a protein, you change the way the protein works, right? So mutating DNA causes an altered function in proteins or sometimes a non-function in proteins. Some of them are non-life threatening, right? Albinism is the result of a DNA mutation that causes a person to not be able to produce melanin, right? So if you can't produce melanin, you don't have any kind of coloration, right? But it's survivable. You wear sunscreen, you're fine, right? Some of them, though, can be fatal, right? Things like Huntington's, right, which is a kind of a neurological degenerative disorder, is a result of this kind of altered protein situation, right? And it results in premature death, unfortunately. One of the first examples of this that was discovered, though, was sickle cell anemia. It's kind of like the famous, like, model example of having a mutation in your DNA that specifically alters the shape of a protein, right, that causes a specific disorder. So like this single mutation causes this specific disorder, right? Kind of end of story. So sickle cell anemia was the first disease shown to be kind of the result of this like single gene, right, single protein mutation. And so it's a defect in the hemoglobin, right, that would carry oxygen in a red blood cell. And so what actually happens, if you have sickle cell anemia, the gene that codes for your hemoglobin creates this kind of misshapen version, right? So normally your red blood cells are these nice little kind of donut shaped guys. But if you have this DNA mutation, your hemoglobin's kind of misshapen in some of these cells, and it causes the red blood cells to kind of be stretched out and have this sickle shape, right? Which is why it's called sickle cell, right? And if your hemoglobin is misshapen, kind of makes sense. It's not as good as it's at its job as it should be, right? So if your hemoglobin is the wrong shape, it can't carry oxygen and deliver it to tissues the way it's supposed to, right? So you are anemic, kind of by definition. Your red blood cells aren't working as well as they could. Now, interesting thing with sickle cell, right? Sometimes these mutations have other effects on our physiology, right? So you have sickle cell, you've got some misshapen red blood cells, you're anemic. As long as you kind of, it is a recessive trait. So as long as you're not kind of overly affected by it, you are frequently okay. Probably. Right. It would affect your, yeah, your ability to kind of, to exert yourself just in general, right? It's a recessive trait, at least in the US, right? It is more common in African Americans. It's more common in African populations generally. This is, if you look at kind of the phylogeography, basically of this DNA mutation, right? If you kind of follow this back in the human family tree, as it were, the mutation arose in Africa where there is a high rate of malaria, which is a very deadly, right, parasite, blood parasite specifically. So the trick is, you guys know anything about malaria? So malarial parasites infect your red blood cells, cause them, you know, fill them up with their parasite cells, cause them to burst. It's bad. But turns out, if you carry this recessive trait and some of your red blood cells are misshapen and still working, right, just not working great, the parasite can't infect misshapen blood cells. So it actually was kind of selected for evolutionarily, right? People who, right, when this mutation happened, people with sickle cell weren't just kind of like selected against. There was actually kind of an evolutionary benefit along with the disorder in a sense, right? Because it provided this resistance to malaria, which is a big problem in, well, in a lot of tropical areas generally, right? So you kind of have this like, I don't know. You're more susceptible. Yeah. It's a blood parasite. It's Plasmodium. I can't remember the name of the little protist, but it's carried by mosquitoes, and it infects the red blood cells and causes them to burst. It's kind of a initially feverish, kind of like fever, chills, aches sort of thing. There are strains that are cerebral that are much more dangerous, depending on depending on what region you are.
So could that be changed by environment, you think? If you were to be brought up in an environment more towards like the city Amazon, where it's more
Like,
likely to happen, would you have more a possibility of that resistance?
so
Or
The resistance is just the result of this like single gene mutation. So that's just, you wouldn't develop it. It would just be, did you inherit it from your parents? And if you did inherit it from your parents, while it does cause you some problems, it would also have this benefit, basically, if you lived in one of these kinds of areas. Yeah. So sickle cell. But there are loads of examples. I think there's a whole table of them in the textbook. But a few others, right, that we may or may not have heard of, right? Cystic fibrosis is one, affects about 1 in 2,500 Caucasians, which is kind of a weird, anyway, we won't get into what Caucasians are. But the mutation, it's complicated, causes your body to be bad at making mucus. Mucus, gross, but is important, right? Lines all of your various like openings and tissues, and your lungs and everything is all full of mucus. But in cystic fibrosis, the mucus you make is too thick. And so you end up clogging your lungs, your liver, your pancreas, right? And it results in kind of just a whole kind of host of issues. It's a recessive trait. So again, it's a recessive trait, something you could carry and not know you have, right? Or at least that you don't know you're carrying the allele for, something you could potentially pass on. Tay-Sachs is another recessive one. This is a central nervous system disorder. And this one is kind of a not survivable one. It deteriorates the central nervous system in infancy. This one specifically is more common in Ashkenazi Jews. So, because over the history of humans, various groups of humans have been very isolated for different reasons, right? So if you have, say, a religious group that is very specifically like, maybe not today, but historically, right? They only married other Ashkenazi Jews. And if a mutation arose in that very closed population, right, it's going to get passed around. And so you end up with some of these genetic disorders being much more common in certain kind of people groups, whether it's regional, right? It can be geographic, right? If you have a group of people who've been living on an island for thousands of years and not kind of affected by other things, or if for some other kind of like behavioral choice reason have been very isolated, right? These disorders can kind of become specific to a group of people, whether it's because of the way those people kind of conduct themselves or they're secluded in some given region for a long period of time, right? So yeah, so Tay-Sachs is one of those things. Huntington's disease, not really specific to any particular region or people. I don't think that's where it came from, but it's a dominant trait. It's a not good one. Causes brain tissue deterioration in middle age. Yeah, it's not what you want. But that's the thing, right? You can have it and have kids and give it to them and then find out later that you have it, right? So you don't live past 40, but if you've already had kids, then the genes go on, because that's how genes do, right? Your DNA really only cares if you reproduce. If you die immediately after, it turns out your genes don't care so much. From an evolutionary perspective, right? The trait gets passed on. So, but there are lots of examples. These are just a few of the kind of more, I don't know, kind of big names, I guess, in genetic disorders. All right. Last set of kind of interesting genetic stuff is nondisjunction. So nondisjunction is the failure of analogous chromosomes or sister chromatids to separate in meiosis, right? So in meiosis 1, we've got these double copies of homologous chromosomes, and theoretically, they should get sent to different places. Sometimes there's a mistake and both of them go to the same place. Same thing in meiosis 2. You've got these sister chromatids, I think more common in meiosis 2. You've got these sister chromatids, and theoretically, they should get split and sent different places, right? One copy to each cell. Sometimes, though, something goes wrong and both copies get sent to one cell and the other cell doesn't get a copy, right? So we call this aneuploidy, right? This gain or loss of a chromosome, right? That is what happens when non-disjunction occurs. In autosomes, this is usually kind of a no-go, which kind of makes sense, right? Your chromosomes are all coding for lots of stuff. If you're missing a whole chromosome worth of genes, that's not really, that generally those cells just don't develop, right? The result we either call usually monosomy or trisomy, right? You should have 2 copies of every chromosome. If you're missing one, then you've got monosomy of some chromosome. If you got an extra one, then you have trisomy, right? You gained a copy. Because we should all, I guess, be disomas in that sense. Nobody says that, right? So, smallest autosomes, you can get an extra one and be okay sometimes. So our smallest chromosomes, 13, 15, 18, 21, and 22. 13, 15, 18, you can be born, but you don't survive very long. 21 and 22, though, you can have an extra copy and survive to adulthood. Right, and this is what Down syndrome is. All Down syndrome is having an extra copy of your 21st chromosome. So it's also called trisomy 21, right? So people are born with Down syndrome, they survive to adulthood, right? There's kind of a set of traits and conditions that go along with that, kind of some medical concerns and things that come from having this third copy. But these people live, relatively normal lives, right? It's not uncommon, right? 1 in 750 children is really not a small number. And it is influenced by the mother's age. So the older you are when you have kids as a female, the larger your risk of having a child with Down syndrome. Risk starts to go up at 35 and then specifically at 40, huh?
That's scary. I don't honestly like. I wouldn't know what to do.
Yeah.
I give credit to all the parents that have to deal with that. I know my cousin. They have 6 kids and 2 of them have Down syndrome.
Oh my goodness.
Yeah.
Yeah, it's a big thing.
So is that more of a higher percentage once you hit that 35, or is it still like, does that say Down syndrome at 21, or is it 3 times?
Your 21st chromosome, you have an extra copy of chromosome 21. And so that tends to be more common, right? Because this is like the 21st, kind of the extra chromosome is associated with the mother's age. Like, it kind of makes sense. As you get older, your cell division and all of your stuff in broad terms gets a little less accurate. So it's just more likely to happen, right? As a woman gets older, she's more likely to have this kind of non-disjunction of that 21st chromosome. She's probably more likely to have non-disjunction at lots of chromosomes, but right, none of those babies are surviving at all, like even to fertilization potentially, right? So you just see that increase with age. All right. But this can also happen in sex chromosomes, right? In the X's and Y's. And these people usually don't experience developmental abnormalities, right? They're generally completely survivable. They have their own kind of associated phenotypic things, which is fine. That happens with any kind of inherited situation. So there's some unique features, but they don't result in death, right? Usually. So, for example, if you have non-disjunction in female gametes, as an example, right? So meiosis has happened, and some of these egg cells have 2 X's, and subsequently some of them are going to have no X's. So if this egg is what's passed on, right, you're going to have a female, right, because the male is passing on an X chromosome. She's going to have 3 X's, which is called triple X syndrome, right? If the mother passes on 2 X's paired up with a Y, you have a male who just has an extra X chromosome, right? Turner syndrome is where the female has passed on no X chromosomes, but the father passed on one. The one non-viable option is if the zygote or the embryo receives no X chromosomes, right? There are genes that you need on the X chromosome, so you got to get at least one. But other than that, it's actually a lot more plastic, right? It's kind of this interesting situation where sex determination is much more plastic than any of our other kind of chromosomal inheritance. But the things that kind of come along with that, so X chromosome non-disjunction, which is kind of what we were just looking at, right? So you can inherit 3 X chromosomes. And basically all that happens, right, we've talked about how this gets controlled basically. you can make 2 bar bodies, right? You just turn 2 of them off instead of turning on 1. I've got 3 X chromosomes, I'm going to turn off 2 and express 1, right? So these people, if anything, are a little taller on average, and that's kind of it as far as phenotypic. Well, probably for multiple reasons, but yeah, right. Yeah, so these are females who are tall. So if a male inherits 2 X chromosomes, you get Klinefelter syndrome. So this is a male, so genetically male, but with some female body characteristics, right? So you kind of actually have a chimeric sort of situation when this occurs, because you're going to express some of those X chromosome traits along with being kind of genetically male. Turner syndrome is where you only inherit 1 X chromosome, so you're just kind of missing one altogether, right? And these individuals they're not able to have children. They tend to be shorter, and they tend to have a webbed neck. They just it's just thick. It looks thick at the sides, right? So you can have Turner syndrome. Y chromosome nondisjunction, a little less common, but it does happen, right? Is where you inherit an X from your mom, and then you get non-disjunct Ys from your father. It's called Jacob syndrome. These people are able to have kids, normal appearance. You wouldn't know. There are people who have done studies and kind of associated this with various like kind of behavioral or aggression issues. It's possible. But yeah, so there's actually a lot of plasticity, turns out, in kind of sex determination in humans. It's not just, there aren't just 2 options, turns out, even at the genetic level, like at the most basic level of things. So you can have this non-disjunction of sex chromosomes, totally survivable. Versus kind of autosomes where it becomes a much more detrimental situation. All right, last one, genomic imprinting. This is fun. Genomic imprinting is where, so you inherit an allele, but the phenotype, right, the thing that happens because of that allele is different depending on which parent you got it from. So if I inherit this trait from my dad, it's going to look different than if I inherit the exact same trait, right, the identical piece of DNA from my mom, right? It'll result in 2 different things. Right? Only happens in mammals and plants. Sure, whatever. So one example is Prader-Willi versus Angelman syndrome. So there's this allele, and if you inherit it from your father, kind of the analogous allele from your mom just gets turned off completely. All right, results in respiratory distress, obesity, short stature, some intellectual disability, and obsessive compulsive disorder. Whereas if you inherit the exact same allele from your mom, the gene from your dad gets turned off, results in delayed development. Again, some intellectual disability, hyperactivity, aggression, and inappropriate laughter, right? And so it's kind of an interesting thing where the exact same piece of DNA code will do different things depending on which parent you get it from, right? And this is called epigenetics. So this is called epigenetic inheritance, where it's a thing you inherit. It is mitotically or meiotically stable, right? It's not like some weird thing that like happened to you in development. It's a thing that you can pass on, that your cells can divide and pass on, right? But it doesn't actually have to do with the DNA code. It's like something else. Epi means like above or on top. So it's like something on top of your DNA coding, right? That's affecting how these traits are inherited. This is kind of another big area of kind of scientific research these days. It's very chemistry-ish. I find it interesting, but I don't read a whole lot about it, to be honest. It has to do with like, people look at things like methylation of the various groups hanging off your DNA. There's all kinds of weird extra chemical stuff that can be going on that can affect kind of how these traits play out, I think it's interesting. All right, so you can have epigenetic inheritance, right? Kind of inheritable DNA things that are like kind of beyond the code itself. All right, so now that we're all terrified of all the horrible genes that we might be holding, right? There are things that people do, right? Like if you, well, either you can just have cats, this option, or if you decide you want to have kids, or if you just want to know kind of your own condition, like what might happen to you in older age, what sort of things you might be carrying that you don't know about, right? There's all kinds of genetic counseling, sequencing, and kind of things you can do to kind of learn about yourself and what different genes you might be carrying, either kind of for your own personal information or if you're interested in having children. So Genetic counseling is where you can like identify parents at risk of various genetic disorders, things they might be carrying, right? Recessive alleles they might have they don't know about. Pedigree analysis is kind of an obvious step one, right? It's what we were doing before we knew about DNA. Do you have a grandparent with some condition or disease or disorder or something, right? And if you do, what is the likelihood, right? You can then determine the kind of the probability that you're carrying that recessive allele and would you pass it on to your kid, right? But there are also tests you can do during pregnancy, right? So if you're looking to have kids and you want to know what you might be passing on, there's a couple tests that they can do. Amniocentesis or chorionic villus sampling. Amniocentesis is a bit more of a high-risk procedure. I mean, anything where you're kind of interfering in the fetal space, right, can be a little touch and go. But in high-risk pregnancies where you really think something might be wrong or you really want to know, right, if something's all right, you can actually sample fetal cells from the amniotic fluid around the fetus. They do that at around 4 months. But you can also do chorionic villus sampling, which you do a little earlier, actually right around 2 months. So it's less invasive and it just takes a bit of cells from the chorion. It's just part of the placenta. So they take some cells basically from the placenta that they can then test and kind of see if there are, you can kind of get ahead of anything that might be coming, whether, you know, it's something you just need to be kind of like mentally prepared for or something you can medically do something about. Okay. Somebody's not muted. All right. And that is the end of chapter 13. So we'll do a couple review questions and then we'll finish it up on Wednesday. We won't do this whole shebang. Let's do a couple. We got a couple minutes. I gotta watch. All right. What is chromosomal theory of inheritance? What does its name imply that it is? Something about chromosomes, right?
And that their behavior during meiosis is physical base of inheritance patterns that Mendel observes in the book.
Wow, that was like an internet definition, right? That's not what I said. Yeah, I didn't say that in my words. Yeah, yeah, but that chromosomal theory of inheritance just states that your DNA, your chromosomes, and the way they behave during meiosis results in, right, the genes you inherit from your parents, right? Because we didn't always know that. They thought that like it was like something in your proteins or something, right? But it is your chromosomes. Your chromosomes carry the information that you inherit from your parents, and the behavior of those chromosomes in meiosis affects how you inherit it. Yeah. What about sex-linked genes? What is a sex-linked gene? Just most basic definition. That's sex determination. That's associated with, yes. So a sex-linked gene is a gene which, yeah. Yeah. It's also called X-linked because it's a gene.
That's linked with the X chromosome.
Yes, it is a gene found on the X chromosome, right? So sex-linked or X-linked genes. So how are they then inherited? Well, from either, but it will affect males disproportionately because what? Because they're, well, so if you're a male, why would you be inordinately affected by a gene on the X chromosome? More so than a female. Because you only have one. By default, if you're genetically male, anything on the X chromosome becomes a dominant trait because you've got nothing to balance it with, right? So things like hemophilia, things like red-green color blindness, right? If you are genetically male, you only need to inherit one of those alleles and you've got it, right? Whereas if you're a female, you can carry it as a recessive trait and not have hemophilia, not be colorblind, etc. All right. That is time. We'll finish up that review on Wednesday.